Good morning, everyone. Today we will be talking about the stages of human embryo development. Human development begins from fertilization and continues until birth. The embryonic period is the earliest and most critical phase of development because all major and body system starts forming during this time. Any disturbance, genetic error, or exposure to harmful factor during this stage can result in congenital abnormalities. Therefore, understanding embryonic development is very important in biology, medicine, and reproductive science. Embryonic development. This slide explains the definition of embryonic development. Embryonic development refers to the early growth and differentiation of a human after fertilization. It starts with the formation of zygote and continues until the end of 8th week of pregnancy. After the 8th week, the developing organism is called a fetus. During the embryonic period, rapid cell division, cell differentiation, and organ formation occur, making it the most crucial phase of prenatal life. Gametogenesis. In this slide, gametogenesis is explained. Gametogenesis is the process by which male and female reproductive cells are formed. In males, spermatogenesis occurs in the testes and produces sperm, while in female, oogenesis occurs in ovaries and produces ova. These gametes are haploid, meaning they contain 23 chromosomes. Gametogenesis is essential because it ensures proper fertilization, maintains chromosome number, and introduces genetic variations in the offsprings. Fertilization. This slide explains fertilization. Fertilization is a process where a sperm fuses with an ovum, usually in the ampulla or the fallopian tube. The fusion forms a zygote and restores the diploid chromosome number of 46. Fertilization also determines the genetic sex of the baby and activates metabolic and cellular process necessary for embryonic development. It marks the beginning of a new human life. Zygote stage. Here the zygote stage is explained. The zygote is a single cell diploid structure formed immediately after fertilization. It contains complete genetic information from both parents. Although the zygote does not increase in size, it undergoes rapid mitotic division while moving through the fallopian tube towards the uterus. This stage is very short but extremely important for further development. Cleavage. Cleavage is a series of rapid mitotic division of zygote resulting in smaller cells called blastomere. During cleavage, the overall size of embryo remains unchanged because division occurs within the zona pellucida. Cleavage increases the number of cells and prepares the embryo structurally and functionally for implantation in the uterus. Morula stage. The morula is a solid ball of about 16 to 32 blastomere. It resembles a mulberry in appearance, which is why it is called morula. This stage usually occurs around the 3rd or 4th day after fertilization. At this stage, the embryo enters the uterine cavity and continues further development. Blastocyst formation. The morula develops a fully filled cavity and transforms into a blastocyst. The blastocyst has two main components, the inner cell mass and the trophoblast. The inner cell mass develops into the embryo while the trophoblast forms the placenta and the supporting structure. This stage is very important because only a healthy blastocyst can successfully be implanted. Implantation. It is the process by which the blastocyst attached to and embeds into the uterine endometrium. It usually occurs around the 6th or 7th day after fertilization. During implantation, the trophoblast invades the uterine lining and starts forming the placenta, which enables nutrient, oxygen, and waste exchange between the mother and the developing embryo. Bilaminar germ disc stage. The inner cell mass differentiate into two layers called the epiblast and hypoblast. This stage occurs during the 2nd week of development. The amniotic cavity forms the epiblast and the yolk sac forms the hypoblast. This structure plays an important role in protection, nourishment, and early development. Gastrulation. It is one of the most important stages of embryonic development. Gastrulation is the process by which the bilaminar germ disc is converted into a trilaminar germ disc. During this stage, 3 primary germ layers are formed. Gastrulation establishes the basic body plan and body axis. Any defect during gastrulation can lead to several development abnormalities. Now the 3 germ layer. First is the ectoderm, which forms the skin, nervous system, and the sense organ. The mesoderm forms the muscle, bone, heart, kidney, and blood. The endoderm forms the digestive tract, lungs, liver, and pancreas. All organs and tissues of human body originate from these 3 germ layers. Neurulation. Neurulation is the process by which the ectoderm forms the neural tube. The neural tube later develops into the brain and spinal cord. Neural crest cells also arise during this stage and migrate to form structure like peripheral nerves and facial tissues. Neurulation is essential for proper nervous system development. This slide discusses somite formation. Somites are paired, segmented blocks of mesoderms found on either side of neural tubes. These somites later differentiate into vertebrate, skeletal muscle, and dermis of the skin. Somite formation gives segmentation and organization to the body structure. Organogenesis. Organogenesis is the stage during which major organs such as the heart, brain, limbs, liver, and kidney began to form. It occurs between the 3rd and 8th week of the development. This stage is highly sensitive to harmful agents like drugs, infection, alcohol, and radiation, which can cause congenital malformation. Extraembryonic membrane. This includes placenta, amnion, chorion, and the yolk sac. The placenta allows the exchange of nutrient, gases, and waste. The amnion surrounds the embryo with protective fluid. The chorion contributes to placental formation, and the yolk sac helps in early nutrients and blood formation. Embryo to fetus transition. At the end of the 8th week, all major organs are formed. Although they are not fully functional, from the 9th week onwards, the developing organism is called a fetus. The fetal period mainly involves growth, maturation, and functional development of organs. This slide summarizes the entire topic. Human embryonic development is a step-by-step, continuous, and highly regulated process. Each stage plays a vital role in forming a healthy individual. A proper understanding of embryonic development is important in medical science, reproductive biology, and the prevention of birth defects. ART and IVF, that is fertilization stage. This slide links embryology with assisted reproductive technology. In IVF, fertilization occurs outside the human body in a controlled laboratory environment. The fusion of sperm and ovum is carefully monitored by embryologists. This technique helps couples who are unable to conceive naturally due to infertility. Cleavage and blastocyst. Embryo development during IVF is explained. After fertilization, embryos are cultured in the laboratory and observed through cleavage and blastocyst stage. Embryo grading is done to identify the healthiest embryos. Selecting high quality embryo improves implantation rate and overall IVF success. Implantation support. This slide explains supports in IVF. Hormones such as progesterone are administered to prepare the uterine lining. Proper endometrial thickness and receptivity are maintained to support embryo implantation. These measures significantly increases the chances of successful pregnancy in IVF treatment. Thank you. I hope you like my presentation.